she can eat anything, she has a fast metabolism, or our metabolism slow as we age. That's why we gain weight. But do you really know what exactly our metabolism do? Even while we at rest, our metabolism does not stop. It constantly provides energy for basic body functions like breathing and digestion. For example, after we eat anything, our digestive system uses enzymes to break down proteins, lipids, and carbohydrates into amino acids, fatty acids, and simple sugars. Assalamualaikum and hi student! I'm teacher Aisha and today we are going to learn about chapter 5 Metabolism and Enzymes. So, what is metabolism? Metabolism refers to all the chemical reactions that occur in a living organism involving the conversion of food into the energy in the form of ATP and the formation of carbohydrates, proteins, lipids, and nucleic acids. These processes are essential for the growth, reproduction, and the maintenance of the structures of living organisms. Let's look at these two types of metabolic processes, which are catabolism and anabolism. Catabolism is defined as process of breaking down complex substances into the simple substances. For example, here we see a complex substance made up of many atoms. So when it's broken down to form the simple substances, then this reaction releases energy because the chemical bonds are broken. An example of catabolism is the breakdown of glucose during cellular respiration to generate energy. In contrast, anabolism is defined as the process of synthesizing complex molecules from simple molecules. Let's look at this example here. So, when we have a lot of simple molecules and when we combine them together so that they form bonds among each other, then a complex molecule has been synthesized. Usually, enzyme is needed to form the chemical bonds, so this reaction absorbs energy. An example of anabolism is the formation of glucose during photosynthesis. In this subtopic, we are going to explore the world of enzymes. What is enzymes? Enzyme is an organic catalyst by the cells of living organisms. Well, enzyme has a specifically shaped area called an active site, where items can bind called substrates. It's very specific binding because the active site is specifically shaped for the substrate that binds there. Very specific. Enzymes are a fascinating group of chemical substances in our human body, which has ability to release speed reactions. Let me give you a great real example, the lactase enzyme. Another really cool thing about enzyme is that they often ending ASE like lactase, while sugar ending OSE. So, to name an enzyme, if the substrate is lactose, add ASE to the name and remove OSE from the name. Then, the enzyme that catalyzes the hydrolysis of lactose is named lactase. Let's move on to the characteristics of enzymes. The first is enzymes act rapidly. The second is enzymes are only required in small quantities and are reusable, as can be seen from the log and key hypothesis. The third is the structure of enzymes remain unchanged and are not destroyed after a reaction. The fourth is enzymes are biological catalysts that speed up biochemical reaction. Next, the reaction of enzymes are specific, meaning they can only act on one specific substrate to catalyze one reaction. This is because each enzyme has an active site with a specific configuration. Some enzymes require cofactors such as vitamin P and magnesium ions to work more efficiently. Enzyme activity can be slowed down or stopped by enzyme inhibitors. Lastly, most reactions catalyzed by enzymes are reversible. Now, classification of enzymes. So enzymes can be divided into two types based on where they function. These are intracellular enzymes and extracellular enzymes. Intra means inside, which means these enzymes are synthesized in a cell for its own use. Whereas extracellular enzymes, the word extra means outside the cell. So these enzymes are synthesized in the cell, but they are secreted out the cell. Okay, so moving on, what's next on our list? Oh yes, mechanism of enzyme action using the lock and key hypothesis. So just a quick review, let us look at this animation of mechanism enzyme action. Firstly, the specific substrate approaches the enzyme. Next, substrate combines with the active site to form the enzyme-substrate complex. Now, once the enzyme-substrate complex is formed, a reaction takes place and a product is formed which then leaves the active site as the reaction is complete. Alright, in this video, we are going to look at the mechanism of enzyme action and factor changes. So, these are four factors that affect the rate of enzyme activity. There are temperature, pH, substrate concentration, and enzyme concentration. So, let's start with the first factor, temperature. At low temperatures, enzymes catalyze reactions. However, when temperature rises, the kinetic energy of substrate molecules and enzymes increases. This increases the frequency of effective collision between the substrate molecules and enzyme molecules. With every 10 second Celsius increase in temperature, the rate of enzyme-substrate reactions doubles. At optimum temperature, the enzyme reaction is at its maximum. After reaching the optimum temperature, any further increase in temperature will reduce the enzyme activity rapidly, where enzymes become denatured as the chemical bonds in the enzyme molecules break at extreme temperatures. The second factor, pH. Enzyme activity is influenced by the pH of the solution, with most enzymes being most effective at the optimal pH range of 6 to 8. For example, the salivary amylase works at pH 6.8. However, there are some exceptions. For example, pepsin enzymes in the stomach act optimally when the pH is between 1.5 and 2.5. The duodenum trypsin enzyme, on the other hand, works well in an alkaline environment with a pH of roughly 8.5. Next factor is substrate concentration. When the concentration of enzymes remains constant, while the concentration of substrate increases, the reaction rate controlled by enzymes increases, resulting in an increase in products. This is because, as substrate concentration increases, the opportunity for effective collision between the substrate and enzyme molecules also increases. The reaction rate remains constant until the maximum level, at which enzyme concentration becomes a limiting factor and all active sites are saturated with substrate. The last factor is enzyme concentration. The rate of enzyme concentration increases with enzyme concentration due to more active sites that are ready for catalytic action. If enzyme concentration doubles in one reaction, the amount of substrate converted to product also doubles, provided there is an excess supply of substrate. At maximum rate, substrate concentration becomes the limiting factor, and adding more substrates can only increase reaction rate. Moving on to the last subtopic, enzymes are widely used in the commercial sector and at home in daily life. They are extracted from natural resources like bacteria and can be produced synthetically. Enzymes like amylase and protease are used in the food processing to improve the quality. Pectinase is used in fruit juice production to clarify and break down pectin in order to improve juice extraction. Enzymes like protease and lipase are used in laundry detergents to break down protein and lipid paste stained on clothing. In the leather industry, trypsin is used to extract fur from animal hide to make leather products. So that's all from me. Stay tuned and see you again in the next video. Bye!